Good day to everyone, and thank you for being here. This lecture is on basic ophthalmic pathology, and the main axes that I'm going to go through are basic cytology, inflammation, immunology, and criteria of malignancy. And to start with basic pathology, as you can see, this is a diagrammatic presentation of the human cell, where the nucleus is the center of the whole thing, with the nucleolus inside, and the nucleus is responsible mainly for cell division. And there are the ribosomes, which are responsible for protein synthesis. And as you know, proteins in the cell are the signaling code for every biological process, including genetics that goes on. And then there are other organelles like the vesicles, and the rough endoplasmic reticulum refers to the structure that makes the skeleton of the cell, and it's rough because it's studded with ribosomes. There is the Golgi apparatus, responsible for many chemical processes that go inside the cell, and the smooth endoplasmic reticulum, which is just a sort of a skeleton that is not studded with the ribosomes. There are vacuoles that are the products of taking in or expelling out structures into and from the cell. And all this is swimming in the cytoplasm, with the lysosomes, which are responsible for the degradation of other products of metabolism and noxious agents. This is the structure of the mitochondrion, and mitochondrion is the singular and mitochondria is the plural, and mitochondria are the power bank, or let's say the engine of the whole cell, and its inside is formed of a membrane that is convoluted so many times to make a very big internal surface. The structure of the tissues of the human body, whether this be the blood vessels or the viscera, is in general formed of an epithelium, an endothelium, and a matrix. For the blood vessel, it's covered from the outside by an epithelium or by the tunica adventitia, and covered from the inside by the endothelium, and in between there is the tunica media, which is the matrix. For the conjunctiva, for example, it's covered from the outside by the epithelium, and there is matrix, and the endothelium here is not presented. The staining techniques in pathology in general are very important to identify and characterize the tissues and the cells. And this here in ophthalmic pathology is through two main stains, which are hematoxylin and eosine. We refer to this as H&E, H plus E, or sometimes HE. Hematoxylin is blue, as the text here, you can see it's blue or light blue. It's alkaline stain, and being alkaline stains basophilic structures, and eosine is a red or a bright pink stain. It's acid in nature and thus stains acidophilic structures. What are the basophilic structures that can be stained by hematoxylin? All structures that contain nucleic acids, such as the ribosomes, the chromatin-rich nucleus, and the cytoplasmic regions where RNA is concentrated. While eosine is, as we said, it's an acidic structure, and thus stains eosinophilic or acidophilic structures inside or outside the cells. And this is represented mainly in most of the cytoplasm and other bodies inside the cell, like Lewy bodies or Mallory bodies. And we should know that RBCs are full of a pigment themselves. RBCs are full of hemoglobin, which is red itself, and it stains even deeper red, more intense when it goes under a hematoxylin and eosine preparation. Other intrinsic pigments in the preparations that we might encounter, like melanin, are yellow and brown. Melanin is when it is very dark, when it is very concentrated, is brown in color, and when it's light, it looks yellow. Mind you, I am making the texts here in the color that I'm talking about. The basal lamina of the cells is PAS stain or silver stain positive, per-iodic acid Schiff, reticular fibers stained by silver stain, and hydrophobic structures that are expelling water tend to remain clear or drop during preparation, like fat globules, for example, appear clear in preparations stained by hematoxylin eosine. However, they are not actually clear. Collagen fibers are very acidophilic and thus stained by the red stain in a hematoxylin and eosine preparation. Under the electron microscope, there are no stains, but light shades, and this is how the collagen fibers appear under the electron microscope. Continuing with basic cytology, cytology is the science of the cell. We come to the blood vessels, and looking at a peripheral blood preparation, the abundance is in RBCs and then some RBCs, and then there are some white blood cells. RBCs are red blood cells. The red blood cells are like a pie that is pie stressed or compressed from the sides, and this makes the surface area bigger for exchange of oxygen with the hemoglobin that is inside. Under the scanning electron microscope, they appear as a biconcave disc or pie. These are the RBCs, of course, shades of light under the electron microscope, no color stain. While the white blood cells or the peripheral blood cells in the blood film are divided into two main categories, which are granulated and non-granulated cells. The granulated cells are acidophils, basophils, and neutrophils, and the non-granulated are lymphocytes and monocytes, as well as dendritic cells. So once more, the peripheral white blood cells are two types. The type 1 is granulated white blood cells, where the cytoplasm is full of granules. The non-granulated type, where the cytoplasm is smooth. Granulated cells are acidophils, where the cytoplasm is full of red stained granules. This is the acidophil. The basophil, on the contrary, the basic cytoplasm is basophil stained blue. And the neutrophils have a granular cytoplasm, not smooth, but it doesn't take neither the red nor the blue stain. As you can see, the acidophils have a bilobe nucleus, basophils have a rather kidney-shaped nucleus, and neutrophils have a multi-lobed nucleus. While non-granulated peripheral blood cells are the lymphocytes, the monocytes, and the reticular dendritic cells. The lymphocytes are characterized by having a very big nucleus and scanty rim of cytoplasm around. Monocytes have a pleomorphic nucleus with basophilic non-granulated cytoplasm and dendritic cells don't take the regular stains, but require a silver stain. Here are the eosinophils with red granulated cytoplasm, hematoxylin and eosine. All these are RBCs. Eosinophil with bilobed nucleus. Basophils are having basic stained or basophil stained granulated cytoplasm with a kidney-shaped nucleus, and neutrophils have multi-shaped or multi-lobed nuclei with cytoplasm that is neither taking the red nor the blue stain. Lymphocyte with a big nucleus taking the major part of the whole cell with scanty cytoplasm around, and its surface, when studied by the scanning electron microscope, is full of cilia. Monocytes have a multi-shaped or pleomorphic shaped nucleus. Now we know that these cells are circulating in the peripheral blood. If you take a sample from your blood, for example, it appears, somebody is asking me to raise the sound. I think this way it's better. Bone marrow stem cells are the cells that settled in the bone marrow coming from the peripheral blood. I'm saying again that if you take a sample from the peripheral blood in your veins or arteries, you find the cells, these cells, but the bone marrow stem cells are rather different. Basophils, when come to the cytoplasm, form the mast cells, and monocytes, the non-granulated variety, form the tissue histiocytes and the dendritic cells. Now, once more, the mast cells, which are the basophils in the peripheral blood, are responsible for allergic reactions, while histiocytes, which are coming from the blood monocytes, have the function of phagocytosis and form the lymphoreticular system and tissue macrophages responsible for a big part of the cellular immunity, while the dendritic cells that are stained by the silver stain are the antigen presenting cells. Now we're finished with the basic cytology, and now we shall shift to inflammation. And it's basic thing that we studied in the undergraduate stage, that inflammation is the body immune response to protect itself. We used to say it's the body response against noxious stimuli that may cause harm to the body. And sometimes this reaction is strong enough to harm the body itself, sometimes. And this reaction might be towards pathogens like bacteria, fungi, viruses, and protozoa, or towards damaged cells from the body itself, or against some other irritants like physical irritants, foreign body, for example, irradiation, or chemicals. And inflammation involves immune cells and chemicals, which are the molecular mediators and the blood vessels that dilate and cause the area to become congested or injected. And the cells involved are the tissue macrophages, dendritic cells, histiocytes, and mast cells that we described. So inflammation is a body response where the cells and chemical mediators are involved with blood vessel reaction, and the cells involved are the macrophages, dendritic cells, histiocytes, and mast cells. And for purposes of a clinical approach, we divide inflammation into acute or chronic. This is on basis of the clinical presentation. Acute inflammation might be suppurative, where pus is formed, or non-suppurative, where no pus forms, or might be chronic as granulomatous or non-granulomatous. Granulomatous might be specific and non-specific. By acute, we mean acute signs and symptoms like redness, swelling, pain, and loss of function of the organ. Suppuration is the formation of pus, and pus here is formed by the dead polymorphs, the dead neutrophils that died in the battle against the noxious stimulus, and no suppuration where the inflammation is not accompanied by pus formation. Chronicity means longer time and weaker response. And this long time response might be in the form of formation of granules, granulomas, by formation of masses, or non-formation of masses. And when masses are formed, they are either specific, like tuberculosis, syphilis, sarcoidosis, and so on, or non-specific, where chronic inflammation cannot be given a reason. During the process of chronic inflammation, multinucleated giant cells might form, and multinucleated giant cells are big cells, of course, by measures of the microscope, where fusion of mononuclear phagocytes under the influence of the inflammatory mediators known as cytokines form, and they are, for example, physiologically found in bones. Osteoclasts, which are the bone resorbing cells, are by definition multinucleated giant cells. They have phagocytic activity, but less than their precursors, which are the mononuclear cells, and they have a morphological variation that we are going to study now. But this morphological variation probably has got a query significance on the function. For example, we have Langerhans giant cells and we have foreign body giant cells. Langerhans giant cells are characterized by the multi-nuclei being arranged at the periphery like a horseshoe in an arcuate fashion. And the foreign body giant cells, sorry, I have to correct this spelling mistake. So the foreign body giant cells, where the nuclei are randomly distributed, aggregating towards the center. So Langerhans at the periphery like a horseshoe, foreign body conglomerate in the center. Two-tone giant cells, where the nuclei are central and surrounding an eosinophilic, that is a red cytoplasmic zone, and a peripheral pale zone. By pale here, we mean non-eosinophilic, non-basophilic. While the epithelium-derived multinucleated giant cells have features of both Langerhans and foreign body, that is, the nuclei are arranged both in the periphery like a horseshoe and in the center like the foreign body giant cells. These are seen prominently in viral infections, especially herpes simplex and varicella zoster, and these are seen in xanthoma, xanthogranuloma, and fat necrosis. We are going to come back to this when we study skin pathology. These are the Mallory bodies, which are full of eosinophilic granules seen in some degenerative processes. Now we are finished with inflammation. We shall have some talk on immunology and immunoglobulins. By immunoglobulins, which we abbreviate for IG, or antibodies, these are the antibodies, these are proteins. And under the electron microscope, the configuration of these proteins are like a Y shape with a long arm and a short arm. Each side of the Y is formed of two arms, one long and one short. And at the periphery of the two long and short arms, there is the specific antigen binding site where antigen antibody union forms, and the antigen antibody complexes start in the reactions that result in release of mediators and so on. These polypeptide chains, or sort of proteins, are characterized into four categories: the IgG, IgA, IgE, and IgM. IgG is the polypeptide immunoglobulin responsible for the primary immune response, and the M is for the secondary immune response. IgA is responsible for the hypersensitivity reaction, and IgE is the reagenic hormone, where the atopy is a character. Now we know that the antibodies are normal finding in the human body, and a single cell of the lymphocytes secretes a single antibody, which is the immunoglobulin. If this single cell becomes a group of cells that is cloned, that becomes a sort of a cell culture, this may result in a secretion of a clone of antibodies as well, and this is where the monoclonal antibodies are secreted. MAB, monoclonal antibodies, the suffix MAB, which we read in many drugs that are recently being described, like bevacizumab, ranibizumab, and so on. So a big population of of lymphocytes secretes a big amount of antibodies coming of the same clone of cells, are called monoclonal antibodies. And these monoclonal antibodies can be synthesized, can be stimulated from an animal or a human body. When they come from the mice, we call them OMAB, or we give them the suffix OMAB. So murine monoclonal antibodies have the suffix OMAB. They are responsible for allergic reactions and anaphylactic shock. And when they come from human or human only, they are called, or they are having the suffix UMAB. This is transferring human Ig genes into murine genome, mouse vaccinated against desired antigen, and human IgG produced. When they are chimeric, that is a combination of an animal and human, is like a chimer, which is the fictional monster. They are giving the suffix XEMAB or ZUMAB. Let's say that again. Monoclonal antibodies coming from the mice are given the suffix OMAB, bevacizumab. Chimeric and humanized monoclonal antibodies are given the humanized means synthetic, given the suffix XEMAB or ZUMAB. That is mixing the murine and the human antibodies, 95% human, and they have a very big antigen binding capacity, and thus probably have a higher efficiency in clinical action, while human Ig genes that are cloned in the mice are purely human still and have the suffix UMAB. So human U, murine O, chimeric X and Z, and you may remind yourself of zombies being chimers and zombies. That's why they're giving the X and Z suffix. In the market, there are many monoclonal antibodies approved by the FDA, but for us so far, especially in our country, we have bevacizumab and ranibizumab. You know, bevacizumab is the Avastin that is approved for treatment, I mean by the FDA, approved for treatment of age-related macular degeneration, but off-label. That means not given as a primary drug for this indication. They are mainly indicated for colorectal cancer, and ranibizumab is primarily approved by FDA for treatment of age-related macular degeneration. We described immunoglobulins, we described monoclonal antibodies, and now we come to the cluster of differentiation. What is the cluster of differentiation, or CD? A CD for the cell is the ID for us. My ID number is unique for me, and the CD is the ID of the cell. It's a cell surface molecule acting as a receptor, and it functions in cell signaling to alter cell behavior. Many conferences have been held, and the CDs have been numbered up to 350 so far. And it was even the CDs were given in the first place to lymphocyte cells that secrete certain monoclonal antibodies or have a certain HLA typing, human leukocytic antigen, and this structure or this cell surface molecules receptors have been expanded to many other cells. A stem cell that gives granulocytes, monocytes, lymphocytes by both types, thrombocytes, have a characteristic CD numbers of these 350, and some cells share some CDs, but after all, it is, for example, when we say CD 45 and CD3, that means a T lymphocyte. When we say CD 8, that means a cell that has been altered by AIDS. The CD markers, as I said, are unique for the cells, and these are examples of these cells with their CDs. For us, we should know that a CD is the cluster of differentiation, and it is the cell ID. The commonest are the CD4 and CD8. CD4 is the helper T cell, and CD8 is a cytotoxic T cell. Then dendritic cells express a high level of CD8. These are, as you remember, the antigen-presenting cells. HIV virus binds CD4 on T helper to gain entry, and CD4 and CD8 T cells in blood. That means HIV is going on as infection. Finally, in this lecture, after we have finished the basic cytology and the inflammation and immunology, I am going to talk to you about the criteria of malignancy. And criteria of malignancy can be seen either macroscopically or microscopically. Macroscopically, using our naked eyes, rapid growth is a character of malignancy. Abundance of vessels, blood vessels, is again a character of seen by the naked eye, a character of malignancy. Hyperpigmentation, a lot of pigmentation of different types. Growth around the main mass, that is satellite growth. This very rapid growth that outweighs the vasculature leads to necrosis and ulceration. So macroscopically, the characters of malignancy are rapid growth, hypervascularity, hyperpigmentation, satellite growth, necrosis, and ulceration. While microscopic characters of malignancy are hyperchromatism of the nuclei, where the chromatin strands inside the nucleus are very dark, appearance of multiple nucleoli, small nuclei inside the nucleus, appearance of spindles of mitotic figures, and loss of polarity. And by loss of polarity in malignancy, we mean that the usual line of cells is lost. For example, if a white blood cell divides to form another white blood cell, we find that it divides to form a cell that is not similar to the original cell, and this is what is called loss of polarity. And just to now have a sort of a joke, as you can see, iPhone started by being like a normal cell, and then it hypertrophied, and hyperplasia occurred, dysplasia occurred, and finally so many cameras just resembling cancer. So I thank you very much for attending this lecture, and now you can unmute yourselves if you'd like to ask any question. Please unmute yourself and give us your name and ask your question. No questions? Okay, right. So next Thursday, same timing, 11 a.m., we shall be talking about stem